Hello everyone, this is Vanashri Sahasrabudhe. Today going to discuss you about the chemistry of nucleotides. So you know the term nucleic acids, and what are they? The polymers of nucleotides. So this nucleotides is an important component of nucleic acids. So let's see, there are three components of nucleotides. Which are they? A nitrogenous base or a nitrogen base, which contains a nitrogen containing aromatic ring compound, a pentose sugar, a 5-carbon sugar, and a phosphate. This here you can see in the screen is the general structure of a nucleotide. It contains a sugar. This is the pentose sugar in the box. Then to its first carbon with a N-beta glycosidic linkage is attached a nitrogen base, and to its fifth carbon with an ester linkage is attached the phosphate. Now let's understand some terminologies. So pentose sugar and nitrogenous base together form a nucleoside, and a nucleoside with a phosphate is called nucleotide. So what is a nucleoside? It is a nucleotide without phosphate. So we have two types of nitrogen bases, purines and pyrimidines, and the 5-carbon sugar here is a ribose, or it may be deoxyribose. So in DNA we have deoxyribose, and in RNA we have ribose sugar. And we also have a phosphate group, which is nothing but a phosphoric acid. Now let us study each of these components one by one. Let's start with the pentose sugar. So as the name suggests, it is a 5-carbon sugar, it is aldose sugar, and it contains a 5-membered ring form. The 5-membered ring form is called as Furanose. It has a Furanose ring structure. By the way, the other one you know is pyranose, which is present in glucose. It is a 6-membered ring structure. Here we have a 5-membered Furanose ring structure. Now this is important that the carbon numbers of the sugars in nucleic acids are designated by the prime, this prime, and just to distinguish them from the carbons of nitrogenous bases. So here it is the structure of the sugars. This is the ribose sugar, and this is the deoxyribose sugar or 2'-deoxyribose sugar. So at the second position of the sugar, the OH group is replaced by only the hydrogen. So the name is deoxyribose sugar. Now let's study about the conformation of sugar in the nucleic acids. So the 5-member ring structure, any 5-member sugar ring structure can exist in two conformation. One of them is enveloped conformation, and other is twisted conformation. Here we are going to study about the enveloped conformation. So what is there in this enveloped conformation? Out of the 5 members of the ring, 4 are in single plane, and the one is lying outside the plane, either above the plane or below the plane. Now here you can see the structure of ribose sugar, and you can see that the 5th carbon, the 5'C of the sugar, is already outside the plane, is already above the plane of the ring. So now let's see, in this envelope conformation, any of the 5 members can come out of the plane. Now the problem with the oxygen here is, if it comes out of the plane, the entire structure is not sterically favored, and thus it is not present. Then let's talk about this C4'. Now this C4' is engaged in a bond with the C5' carbon, and so its movement is constrained, and so it does not come out of the plane. The C1' carbon is engaged in the bond with the nitrogenous base, so even this does not come outside the plane. Now remaining 2 carbons, out of the remaining 2 carbons, C3' and C2', in the nucleotide structure, only nucleotide structure, when these nucleotides are not present in the nucleic acids, 2 conformations are possible, either C2' or C3', they can be outside the plane. But when the nucleotides or when the sugars, they exist in the nucleic acid structures, this C' is engaged in a phosphodiester linkage. So the only remaining carbon now is C2' carbon. This C2' carbon can show 2 conformations, either endo or exo. So what is endo conformation? When the C2' carbon is in the plane same as that of the C5', that is CH2OH, when both have same plane, both occupy same plane, then it is called as C2' endo conformation. And when this C2' is in the opposite plane as that of the CH2OH group, this fifth carbon, then it is called as exo conformation. So here you can see the exo and endo, C2' exo and C2' endo conformations. So what is present in DNA? DNA C2' endo conformation is present. Now let's study about nitrogenous bases. So two types of nitrogen bases we have, purines and pyrimidines. The purines are two ring structures, so two rings fused with each other. One is 6-membered ring and one is 5-membered ring. This is the general structure of purines. So how to number this purine structure? That is, you start here from this N and you go here in which direction? That is anti-clockwise direction. And these are two types of purines found in nucleic acids, that is adenine and guanine. Now here you can see the number 1, 2, 3, 4, 5 and 6. And now 7, 8, 9 you go clockwise. So now the name of adenine in that way will be 6-amino purine and the name of guanine will be 2-amino 6-oxopurine. Now another type of nitrogen bases are pyrimidines, which are a 6-membered single ring structures. So how do you number these ring structures? The numbering is starting from the basal nitrogen here and it goes in a clockwise direction. So three types of pyrimidines are found. Those are cytosine, which is present in both DNA as well as RNA. Thymine is present only in DNA and uracil is present only in RNA. So how do you name them? The cytosine is called as 2-oxo-4-aminopurine, whereas the uracil is called as 2,4-dioxopurine. So you can see 2 and 4 oxo group are present. So 2,4-dioxopurine, and the thymine is called as 5-methyl. So here is 5-methyl and 2,4-dioxopurine. 2,4-dioxopurine is already uracil. So thymine is also called as 5-methyl uracil. So this is the overview of nitrogen bases. Now let's see about the properties of nitrogen bases. The important property, both the types of nitrogen bases, they show keto and enol tautomeric shifts. So they exist in two structures, tautomeric structures. The tautomeric structures, the tautomers are nothing but the isomers which are formed by the movement of hydrogen atoms. So in lactam or in keto form, the oxygen here is present in keto, or the carbonyl oxygen is present, whereas in enol form the alcohol is present. So that is keto-enol tautomeric shift. This is the keto-enol tautomeric shift observed in pyrimidines, and whereas this is the keto-enol tautomeric shift observed in purines. Usually keto form is more favored over enol form in general. So in our DNA, keto form of both the nitrogen bases is observed. Another property is the dissociable hydrogens present in these nitrogen bases. So here you can see this is the data of pKa values, or the proton dissociation constants at various positions of the nucleotides. They have also given the proton dissociation constants of phosphate, the pK1 and pK2 for phosphate in different nucleotides. So this decides which nitrogen is protonated at physiological pH, and this has a very much significance in base pairing in the DNA structures. Because of these pK values, the base pairing is affected. That we will see in the next part. Now, another property is the UV absorption. As they are aromatic compounds, due to their aromaticity, they show strong UV absorption, and almost all the nucleotides show a strong UV absorption near 260 nanometer. So this is the UV absorption for adenine, this is for uracil, this is for guanine, and this is for cytosine. So here you can see at physiological pH is pH 7, almost all of them, they show characteristic maximum absorption at near 260 nanometer, And this is again an important property of nitrogen bases for detection of nucleic acids. This is important property because nucleic acids are characterized by presence by highest absorbance or absorption maxima near 260 nanometer. Now, how nucleosides are formed? Pentose sugar plus nitrogen bases, they form nucleosides. Now, how are these linked? They are linked via an glycosidic linkage. In the bond that connects anomeric C'1 carbon, the anomeric carbon, to N9 of purine. So here you can see the N9 of purine is involved in the N-glycosidic bonds in case of purine, and the C'1 carbon of ribose or deoxyribose sugar, and N1 of pyrimidine. So here you can see N1 is involved in the glycosidic linkage. This is very important. Now the glycosidic bonds, these are beta glycosidic bonds. So N beta glycosidic linkages are present. And how are they named? They are named, the pyrimidines are named by adding idine, that is idine, as a suffix to the name of the nitrogen base. So it becomes thymidine, cytidine and uridine. And well, you add ocene on the as a suffix for purines. So you have adenosine and guanosine. So when I say adenosine, that means I am talking about the nucleoside. When I say adenine, then I am talking about a nitrogen base adenine. And when I am talking about thymidine, then I am talking about a thymine nitrogen based linked with the ribose sugar, that is a nucleoside. So what about the conformation of nucleoside? So the conformation in this nucleoside, the nitrogen bases, they are planar and they are perpendicular to the glycosidic linkage, but they are in the same plane as that of the sugar. So they are coplanar. So the nitrogen base rings and the sugar ring both are coplanar, but the nitrogen base is perpendicular to the glycosidic bond. Primarily two conformations are possible, syn and anti. And which is sterically favored in which of the nucleosides? For purines and pyrimidines. Let's see it. So for pyrimidines, anti is sterically favored. So anti-conformation is sterically favored. Whereas for purines, they can exist in both conformation. And this is important to note that in case of purines, the rings, the nitrogen base rings and the sugar are not coplanar, but the nitrogen bases are roughly at right angles to the plane of sugar. So these are the conformations. This is the syn-guanosine, this is anti-guanosine, and this is anti-uridine, which is favored. Now when syn-uridine is favored, then it will be present in the opposite direction, and this O will overlap over this O, and because they are coplanar, it will cause a huge amount of steric hindrance, and the resulting compound will not be stable then. So in order to ensure the stability, this pyrimidines are present in anti-conformations. Now let's talk about the nucleotide. The nucleotide to the nucleosides, to the fifth carbon of the sugar of phosphoric acid is esterified, and that is called as nucleotides. How are these nucleotides named? To the name of nucleoside of that particular nucleoside, a monophosphate term is added. So it is nucleoside 5' monophosphate, which is nothing but a nucleotide. So these are the structures of nucleotides. So it is here it is given the symbols of these nucleotides and the names of the nucleosides, what you call nucleosides and how you call them. So these are the nucleosides. Now let's see an overview how the bonds are formed. So here glycosidic bonds are formed by removing this OH and this H, together a water molecule is removed, and from here this H and this OH together a water molecule are removed. This is an ester bond and this is a glycosidic bond. So this is how nucleotides are formed. That's it for today. Thank you.